circDeep, deep learning approach for circular RNA classification from other long non-coding RNA. Mohamed Chaban 1. Robert M. Williams 1. Austin T. Stevens 1 and JuwWon Park 1,2, 1 Department of Computer Engineering and Computer Science and 2. Brin Bioinformatics Core, University of Louisville, Kentucky 40208, USA. To whom correspondence should be addressed. Associate Editor, John Hancock. Received on December 19, 2018. Revised on June 13, 2019. Editorial decision on June 27, 2019. Accepted on July 1, 2019. Abstract. Motivation. Over the past 2 decades, a circular form of RNA, circular RNA, produced through alternative splicing, has become the focus of scientific studies due to its major role as a microRNA. miRNA, activity modulator, and its association with various diseases including cancer. Therefore, the detection of circular RNAs is vital to understanding their biogenesis and purpose. Prediction of circular RNA can be achieved in 3 steps: distinguishing non-coding RNAs from protein-coding gene transcripts, separating short and long non-coding RNAs, and predicting circular RNAs from other long non-coding RNAs, microRNAs. However, the available tools are less than 80% accurate for distinguishing circular RNAs from other microRNAs due to difficulty of classification. Therefore, the availability of a more accurate and fast machine learning method for the identification of circular RNAs, which considers the specific features of circular RNA, is essential to the development of systematic annotation. Results. Here we present an end-to-end deep learning framework, circDeep, to classify circular RNA from other microRNA. circDeep fuses an RCM descriptor, ACNN BLSTM sequence descriptor, and a conservation descriptor into high-level abstraction descriptors, where the shared representations across different modalities are integrated. The experiments show that circDeep is not only faster than existing tools but also performs at an unprecedented level of accuracy by achieving a 12% increase in accuracy over the other tools. Availability and implementation. https://github.com/uoflbioinformatics/circDeep. Contact JuwWon.park@louisville.edu. Supplementary information. Supplementary data are available at Bioinformatics Online. 1. Introduction. Non-coding RNA, microRNA, Matic and McCuneen, 2006, is functional RNA that is transcribed from DNA, but is incapable of being translated into protein. microRNAs can be categorized into 2 groups based on length. Short non-coding RNAs are shorter than 200 nucleotides, and long non-coding RNA, microRNA, are longer than 200 nucleotides. microRNAs have a critical role in several cellular functions involving protein synthesis within a multitude of distinct processes and gene regulation. Mercer et al., 2009, and the Development and Pathophysiology of Disease, Chen et al., 2012. A subcategory of microRNAs, circular RNA, circRNA, has become the focal point of scientific studies over the last 2 decades in a variety of species due to its correlation with a myriad of diseases including cancer. Bachmeier-Hayda et al., 2015, Bird et al., 2010, Cooper et al., 2009, Erickson et al., 2003, Lukiu, 2013, VC the authors 2019. Published by Oxford University Press. 73. This is an open access article distributed under the terms of the Creative Commons Attribution Non-Commercial License, http://creativecommons.org/licenses/by-nc/4.0/, which permits non-commercial reuse, distribution, and reproduction in any medium, provided the original work is properly cited. For commercial reuse, please contact journals.permissions@oop.com. Bioinformatics, 36-1, 2020, 73-80 DOI, 10.1093/bioinformatics/BTZ537. Advance Access Publication Date, July 3, 2019. Original paper. Morel et al., 2006, and its vital function as a microRNA, miRNA, activity modulator, Chen et al., 2015, Mumzak et al., 2013. circRNA is produced by ligating a downstream donor site, 50 splice site, of a flanking downstream intron with an upstream acceptor site, 30 splice site, of a second upstream intron. This process is a unique type of alternative splicing, referred to as back splicing. This is contrasted by canonical alternative splicing, which joins an upstream donor site, 50 splice site, with a downstream acceptor site, 30 splice site, within a single intron and produces a linear configuration of RNA. Recently, There has been a growing number of circRNAs identified. It has been estimated that circRNAs are assembled from over 10% of genes, Lasda and Parker, 2014. circRNAs have a greater stability than linear RNAs due to their structure which excludes 50 and 30 ends. circRNAs are also immune from exonucleosa-mediated degradation. The detection of circRNAs is a vital operation for continued comprehension of their biogenesis and purpose. A substantial amount of circRNAs have been annotated in the transcriptome with the advent of modern experimental technology. Unfortunately, it remains an extensive problem to distinguish circRNAs from traditionally labeled lncRNAs due to the computational complexity of experimental data analysis and the low expression that almost all lncRNAs have. Darian et al., 2012. As of now, only two tools are available for computational categorization of circRNA. The first is circRNA, Pan and Xiong. 2015, which is a computational approach based on a multiple kernel learning framework trained with a variety of features, i.e. graph features, component composition features, conservation score features, features of ALU and tandem repeats, the ORF and SNPs from transcripts. The second is HELM, which extracts identical features and categorizes circRNAs from other lncRNAs by utilizing a hierarchical extreme learning machine, HELM, algorithm with feature selection. Chen et al., 2018. For the dataset proposed, Chen et al., 2015. circRNA reached 0.778 accuracy with 0.554 Matthews correlation coefficient, MCC, and HELM reached 0.789 accuracy with 0.561 MCC. Neither method is perfect, and both have non-negligible drawbacks that could prevent them from being reliably adapted by the bioinformatics community. Neither succeeds at using features that describe the unique structure of circRNA, and both methods use sequence information with trinucleotide frequencies but fail to employ the co-occurrence relationship of trinucleotides. To address the limitations of existing approaches, we propose circDeep, an end-to-end machine learning framework for robust circRNA prediction. In this work, we introduced an innovative feature descriptor that we called reverse complement matching. RCM, descriptor which aims to extract the potentiality of the flanking sequences to the query sequence to make the circularization process. We also propose another feature descriptor that we called ACNN BLSTM sequence descriptor which combines the asymmetric convolution neural network, ACNN, with the bidirectional long short-term memory network, BLSTM, which is able to extract, from each sequence, local patterns as well as the long-range dependencies. These two novel descriptors are fused with a conservation descriptor which is composed of features that contain information about the conservation of a specific sequence among species, as well as conserved motifs. Finally, to fuse different heterogeneous descriptors, we propose a deep architecture to construct the nonlinear representation from different aspects of information sources. 2. The best of our knowledge, circDeep is the first method that uses a deep model for circRNA prediction. We compared circDeep, circRNA and HELM using the dataset from Chen et al., 2015. Figure 1 shows comparisons of results from different statistical measures to compare circDeep, circRNA and HELM for the same dataset proposed in Chen et al., 2015. In this paper, we describe how the improvements in performance were obtained for circDeep over other circRNA prediction tools. 2 materials and methods. In this section, we describe our deep learning framework that integrates different sources of data to predict circRNAs. We describe a method for extracting distinctive representations of different sour CES, which are subsequently integrated using feature fusion learning to predict circRNAs. 2.1 Feature descriptors 2.1.1 RCM descriptor. Many studies support the idea that reverse complement matching in flanking introns and circularization are highly associated. Ivanov et al. 2015. Demonstrated that reverse complementary sequences between introns bracketing circRNAs were notably elevated in comparison to linear controls. It has also been demonstrated that the presence of long inverted repeats, IR, flanking the mouse STI genes results in the creation of the STI circular transcript in cultured cells. Dubin et al., 1995. Zhang and colleagues provide several lines of evidence to support the deduction that circRNA formation is reliant on flanking complementary sequences, either with repetitive or non-repetitive components. Zhang et al., 2014. Consequently, it has been suggested that RCMs, Figure 2a, encourage hairpin creation of the transcript which describes how an upstream acceptor site, 30 splice site, of an upstream intron is fastened to a downstream donor site, 50 splice site, of a flanking downstream intron. A feature like this can be a strong key feature to help our model predict the potential of a sequence to be transformed to circRNA. Therefore, we begin by deriving score H for computing the pre-ance of reverse complementary sequences in flanking sequences. It represents the absolute number of all reverse complement sequences in the flanking sequences. Therefore, for each query sequence S, we take two flanking sequences, each of length L0 base pairs, BPS. We then split the two sequences into k-mers using the sliding window approach. We extract all subsequences of length k with stride S1/4 resulting in L subsequences L1/4 L0 k thorn 1. There are D1/4 4k possible words of length k in the sequences. For each word in the vocabulary, wa, a 1/4 F1, 4 kilograms, we count the number of occurrences in the left flanking sequence a thorn y and the number of figure 1. Comparison of circular RNA prediction tools. circDeep significantly outperforms previous proposed methods. See section 3 for details. 74 M. Chaban et al. Occurrences of the reverse complement to y in the right flanking sequence occur thorn RCWTH. The RCWTH is the reverse complement of the word wa, for example RCIQ eth thorn 1/4 agi. Then the score of the sequence S is given by equation 1, which is the sum of scores ha of all words wa, where ha is the absolute number of occurrences of the word wa, h thorn k, L0 1/4 x 4 ka 1/41 ha, 1 where ha 1/4 of a minute eth thorn L thorn wa, occur thorn RCWTH. The R score h thorn k, L0 can be informative about the absolute number of reverse complement sequences. This can be very useful, according to current models for the production of circRNAs, which posit that RNA secondary structures formed by inverted sequences in flanking introns are necessary elements for circularization. Dubin and colleagues have suggested that at least 400 complementary nucleotides are necessary for circularization in vivo. Dubin et al., 1995. Other research, Hansen et al., 2013, has shown that artificially surrounding an exon with introns containing 800 nucleotides, NT, of perfectly complementary repeats is sufficient to allow circularization, which suggests that long reverse complement sequences promote circularization. Lacking detailed mechanistic models of circularization generally makes the prediction of circRNAs difficult, but, based on some previous studies, we hypothesize that strengthening the hairpin between the reverse complement sequences may increase the likelihood of circularization. Therefore, the availability of a measure describing the longest reverse complementary sequence in flanking sequences to a query sequence for our classifier should improve discrimination between circRNAs and otherlong. In the following pseudocode, we describe our procedure for putting score V that describes the strengthening of the hairpin between flanking sequences. In the results section, we show the effectiveness of such a feature for discriminating circRNAs from otherlong. In order to evaluate the strengthening of the hairpin between flanking sequences, Figure 2b, our method provides a new dynamic programming approach that computes score V for the longest reverse complement sequences while allowing some mismatches and penalizing non-complementary nucleotides. To calculate the longest reverse complement matching between two sequences of length L1 and L2 respectively, we split each sequence into overlapping k-mers of length k with stride equal to 1, so each sequence is represented by y, a 1/4 F1, L1 k thorn 1 gram and Wj, j 1/4 F1, L2 k thorn 1 gram respectively. RCY is the reverse complement of the word y, for example RC ag 1/4 kaku. Let V thorn L1, L2, k be the score for the longest reverse complementary matching between sequences y, a 1/4 F1, L1 k thorn 1 gram and Wj, j 1/4 F1, L2 k thorn 1 gram with allowing some mismatches. In our formulation of the score V, we are allowing some mismatches for longest reverse complementary matching between sequences. If it contains many mismatches, the score V will be low, which reflects flanking sequences less likely to form a hairpin and more likely to form circular RNA. The time complexity of our algorithm to compute V thorn k, L1, L2 is OL thorn 1 L2. We use k-mers for calm putting score v. As a constraint, at least one k-mer is matched between two flanking sequences. However, this algorithm can be extended to eliminate this constraint by simply taking k1 quarter 1. The choice of the hyperparameters to calculate the score v, such as match score, mismatch score, L1 and L2, are based on preliminary experiments. 2.1.2 Conservation descriptor. The fast cons, Mikolov et al., 2013, method is used to give a score ranging from 0 to 1 to each nucleotide based on its conservation level. We gather precomputed conservation scores from the UCSC database, https://genome.ucs.edu. For every exon sequence in every transcript, we average the scores in the exon sequence and then compute the maximum, the average and the median of those averaged scores. If a transcript does not have an exon, the entire transcript is held as a single exon for our computation. circRNAs have a multitude of conserved docking sites for miRNA, such as CIR7, which holds over 70 selectively conserved miRNA target sites, Goldberg and Levy, 2014. This is a result of most circRNAs having almost identical motif sequences. Consequently, we sum the number of frequencies of successive bases whose scores are larger than the specified threshold and then divide the frequency by the whole length of the sequence. We alter the number of successive bases in the scope of 4 through 7, incrementing by 1 and setting a threshold in the range of 0.5 through 0.7, incrementing by 0.1. This gives us a total of 15 features to create our conservation descriptor. 2.1.3 ACNN BLSTM sequence descriptor. In order to extract ACNN BLSTM sequence descriptor, we build a deep learning architecture as shown in Figure 3, consisting of three main components: embedding layer, asymmetric convolutional neural network, ACNN, and bidirectional long short-term memory, BLSTM, network. First, each sequence is converted to a sequence of k-mer indexes, and then we map the k-mers to vectors found at the corresponding. Figure 2. A. Reverse complement matches between flanking sequences promote circularization of RNA. B. Computing the strength of hairpin in flanking sequences allows circular RNA prediction. Calculation of score Thorn K, L1, L2 for phi 1 quarter 1, L1 K Thorn 1 gram V Thorn I, 0 and 1 quarter 0 for FJ 1 quarter 1, L2 K Thorn 1 gram V Thorn 0, J 1 quarter 0 for FGI 1 quarter 1, L1 K Thorn 1 for FJ 1 quarter 1, L2 K Thorn 1 gram FY 1 quarter RCWJ. V Thorn I, J 1 quarter max 0, V Thorn I 1, J 1 Thorn match score else V Thorn I, J 1 quarter max 0, V Thorn I 1, J 1 Thorn mismatch score V Thorn K, L1, L2 1 quarter max Thorn V Thorn I, J where I 1 quarter FG1, L1 K Thorn 1, J 1 quarter F1, L2 K Thorn 1 gram circRNA classification via Deep Learning 75. Index in the embedding matrix. The embedding matrix is pre-trained first with the Word 2-vec model, Mikolov et al., 2013, called skipgram, that computes the co-occurrence statistics of k-mer and learns to project them into a d-dimensional space road. It will be further fine-tuned during the training process. As the convolution layer in our model requires fixed length input, we add each sequence that has a length less than maxln with special symbol at the end that indicate the unknown k-mers, and for those that have a length longer than maxln, we simply cut extra k-mers at the end of these sentences to reach maxln. So now, each sequence is represented as x1 maxln 1 quarter half x1, x2, zmaxln, 2, where xj2 road be the d-dimensional word vector corresponding to the jth k-mer in the sequence. A deeper network will generally have more representational power than a shallower network, but the training time and the greater number of parameters makes them difficult to train. The number of parameters can be greatly reduced with a minor loss in performance. By the use of asymmetric convolutions, circDeep et al., 2015, allowing for deeper models to be trained using the same resources. We divide the more common KD rectangular convolutional filter into two separate steps. First, we apply N1D convolutions, fall load by NK1 convolutions, circDeep et al., 2015. The 1D convolution filter MJ2 road is applied to each k-mer xj in the sentence and generates corresponding feature MJ. MJ 1 quarter FW1 8 xj Thorn B, 3 where 8 is element-wise multiplication, B is the bias and F is a nonlinear function. We chose the ReLU activation function, because it is known to perform well in CNNs. We get the feature map M2 RL M1 quarter half M1, M2, milliliter, For the K1 convolution with filter W2 and RK is applied to a win DAO of K features in the feature map M to produce the new feature CJ. And the feature map C CJ 1/4 FW2 8 MJ Jth K1 Thorn B, 5, C 1/4 half C1, C2, C Lkth 1, 6 dropout is then employed, Shrivastava et al., 2014, to reduce overfitting. The output of the ACNN is read by a BLSTM, enabling long-term dependencies to be captured in both directions. Recurrent networks are especially well suited for finding dependencies and complex relationships in sequential data. However, they can only recognize very short-term dependencies. This problem is overcome using forget gates, which allow some information to be preserved for long stretches of time while selectively forgetting data that is not needed. In this task in particular, dependencies in both directions are important, so a BLSTM network is used to capture this. 2.2 Feature fusion learning for circular RNA prediction. Since we have different heterogeneous descriptors from different sources of data, feature fusion learning is incorporated in the model. To learn shared features across different sources of data, the proposed model can learn and combine high-level heterogeneous representations simultaneously. The first step in our method involves training the model described in section 2.1.3 and computing the optimal parameters W1, W2, WH, WH1 where H is the number of hidden layers for ACNN BLSTM architecture. Next, for each sequence, we compute the ACNN BLSTM sequence descriptor using the feed-forward pass procedures by taking the final representation before the output layer. We extract, at the same time, conservation and RCM descriptors using methods described in 2.1.1 and 2.1.2. The second step of our method involves teaching the model with the 3 obtained descriptors. To achieve this, we can utilize either late fusion, decision fusion, or early fusion, brute force feature concatenation. Early fusion takes features from the 3 modalities, chains them together, and then uses the new vector form to train a deep neural network. The only issue with early fusion is that it fails to consider the different statistical properties of the Informa tie-in it concatenates together. Late fusion creates a final prediction, which is simply an amalgamation of each unimodal prediction from the 3 modalities. Each unimodal prediction has an output interpreted as a confidence score. These scores are combined to give us the final confidence score. The final prediction allows us to maximize the capabilities of each unimodal classifier. However, this method loses some correlation in multidimensional space. To avoid this, we use a slight variation of late fusion called feature fusion fine-tuned, which is shown in figure 4. In our variation, we put each information source through multiple layers to construct a high-level representation of each source. The final representation is just an amalgamation of each individual high-level representation. While feature learning, individual DNNs are trained beforehand separately and then fused together for the last common training, which utilizes backpropagation. Learned parameters from each model are adjusted automatically during each training epoch. After multiple training epochs, the model can recognize depictions from the RCM, the ACNN BLSTM sequence and the conservative descriptors for ensuing categorization. Additionally, the model is better able to learn features for each modality using backpropagation when various modalities are present. This method avoids various problems present in other methods of fusing, such as struggling to recognize highly nonlinear relationships, superfluity and reliance between several descriptors, and overfitting. 3 results in discussion 3.1 experiment setup. In order to gauge the capabilities of our deep learning model, we used human circRNAs from the database circNIB, Hall, 2000, which figure 3. The flowchart of proposed circDeep for classification of circular RNA from otherlong. It extracts 3 descriptors and then use feature fusion learning to integrate different representations for classification. 76M JuwWon et al. Contains 32,914 human circRNAs carefully selected from diversified sources. After we removed circRNAs shorter than 200 nucleotides, we were left with 31,939 circRNAs to act as our positive dataset. We used GenCode, Zhang et al., 2016, to create our negative dataset, which was made of other LNCRNAs such as process transcript, antisense, LNCRNA, sense intronic and sense overlapping. The annotated LNCRNAs in GenCode have 3 validation levels for RNA annotate tie-in: validation, manual annotation and automated annotated. Only transcripts that were validated or manually annotated were chosen to obtain 19,683 samples that compose our negative data. We then divided each dataset into training data, validation data, and testing data where 75% were used for training, 10% for validation and 15% for testing. The training data was used to fit the optimal parameters for our model. We then used the validation data to test the performance of the model with these parameters. The test data was then used to test the model with the best performance on the validation data. The testing was used to provide an unbiased evaluation of a final model fit on the training dataset. For the unsupervised training of k-mer embedding, we generated the corpus of k-mer sequences by setting k to 3, and the stride s to 1. Consequently, the k-mer vocabulary size was v1/4 43. 1/464. Word 2 vec, skip gram with negative sampling, was implemented using efficient multicore implementation by Jensen, Tatimer and Willis, 2017, an open-source Python library for processing large amounts of text with a focus on topic modeling. We set the window size, context, to 18, the length of the dense vector to represent each trinucleotide to 40, the initial learning rate to 0.025 and the number of iterations to 100. circDeep is implemented in Python using Keras 1.0.4, which is found at https://github.com/fshalit/keras, with the backend of Theno, 0.9.0, Legnini et al., 2017. In order to benefit from the parallel computation of the tensors, we trained our model on a NVIDIA Tesla C2050. To allow the deep architecture to extract an ACNN BLSTM sequence descriptor, we set max length, maxlen, to 8,000 basis points, the number of asymmetric convolutional filters to 100, the filter length to 7, the memory dimension to 100, initial learning rate to 0.02, batch size to 128 and the maximum number of epochs to 45. See supplementary table S1 for more details about architecture. This model is optimized using the RM-SPROP algorithm to learn all model parameters, including the convolution filters. For the feature fusion learning, the number of hidden units for 2 fully connected layers, FCL, for each descriptor are listed in supplementary table S2. Batch normalization is added to all hidden layers. We set the initial learning rate set to 0.01, batch size to 64, the dropout rate to p1/4 0.3 and the maximum number of epochs to 70. The evaluation measures used in the analysis included accuracy, sensitivity, specificity, F1 score and Matthews correlation coefficient, MCC. 3.2 Efficacy of RCM descriptor to explore whether the new proposed features HK, L0, and VK, L1, L2, are able to predict circRNAs and improve the performance of our model. We used Pearson's correlation score, Hall, 2000, to measure the relevance of these features and the dependence between the features and the class label, circRNA or otherlong. In other words, Pearson's correlation score can be used to measure the capability that HK, L0, and VK, L0, features can discriminate between circRNAs and otherlong. It is one of the most powerful feature selection techniques, and it is easy to compute and interpret. The resulting value lies in -1, 1, with -1 meaning perfect negative correlation and Thorne 1 meaning perfect positive correlation. Higher scores equate to more relevant features. We calculate HK, L0, for k in the interval, 3, 11, with stride of 1 and L0 in the interval, 250, 2000, with a stride of 250. We calculate VK, L1, L2, by taking the same length for flanking sequences L11/4 L21/4 L0 and varying it in the interval, 250, 1750, with a stride of 250, and k is varied in the interval, 1, 7, with a stride of 1. We set the matching score to 12 and the mismatch penalty to -2. The results for Pearson's correlation for our proposed features H and V are shown in Figure 5. To make our results more rigorous, we also calculate the information gained for the same features, supplementary tables S3 and S4. We can see that the results are very similar and give the same interpretation using any of the statistical methods. As can be seen in Figure 5, the Pearson's correlations for HK, L0, and VK, L0, are considerably high, starting from 500 basis points in the flanking sequences, indicating that the RCM that promotes the hairpin for circRNAs is mostly located after 250 basis points. The results provide preliminary evidence to support our hypothesis that the features HK, L0, and VK, L0, for flanking sequences. Figure 4. Graphical illustration of our end-to-end deep learning architecture used to extract ACNN BLSTM sequence descriptor circRNA classification via Deep Learning 77. Longer than 500 basis points facilitate the identification of circRNAs since most Pearson's correlation scores are higher than 0.70 and can reach approximately 0.83. It can be seen also from Figure 5 that Pearson's correlation scores increase when the length of flanking sequences is increased for all values of k-mer length. However, these scores are not extracted for larger k-mer sizes and longer flanking sequences because the run time for both features, HK, L0, and VK, L0, is increased with the increased length and size. See supplementary tables S3 and S4. When we compare features H and V for any fixed length of flanking sequences from Figure 5, HK, L0, features reached better Pearson's correlation scores than VK, L0, features. This shows that the absolute number of RCM is more informative than the score of the longest hairpin in the flanking sequences when determining the likelihood of circRNA formation. Therefore, our results strongly indicate that circularization can be altered by the competition of RNA pairing across flanking introns and does not rely on the single long-just reverse complement sequence. Since we aim to obtain a robust classifier, we ranked the features using the Pearson's correlation score and then selected the first 70 ranked features to obtain the final RCM descriptor composed of 42 HK, L0, features and 28 VK, L0, features. 3.3 Efficacy of ACNN BLSTM sequence descriptor 3.3.1 Comparing with baseline architectures for sequence classification to evaluate the efficacy of our proposed ACNN BLSTM sequence descriptor, we compared the performance of our proposed architecture, Figure 3, with several baseline methods for sequence classification. We compared with a one-hot CNN architecture adopted by Zhang et al., 2016, a one-hot CNN LSTM adopted in Tatimer and Willis, 2017, a glove CNN BLSTM adopted by Min et al. 2017, a one-hot CNN BLSTM, a one-layer LSTM, a one-layer by LSTM, an ACNN and a CNN that we implemented. See Supplementary Table S5 for more details about each architecture. Table 1 shows the accuracy on training and validation sets and the accuracy and MCC on the test set for all methods described above in addition to the number of epochs and average running time per epoch. By comparing different models, we notice that convolutional neural networks, one-hot CNN, ACNN, CNN, outperform LSTM-related models, LSTM, BLSTM. This validates the importance of convolutional operations in predicting circRNAs. The ACNN architecture is considered the fastest and the best among convolutional neural networks. It reached 0.833 accuracy and 0.6625 MCC on test data, which demonstrates its power for capturing local sequence patterns and detecting spatial motifs that characterize the circRNAs. For LSTM-related models, BLSTM architecture achieved best performance with 0.8020 accuracy and 0.5997 MCC. This indicates that it is more useful to have access to both the past and the future context for predicting circRNAs. The most interesting aspect of our results is how the performance is improved by combining a convolutional neural network with a recurrent neural network. This is not surprising when considering that recurrent neural networks excel at sequential modeling while convolutional networks completely fail at learning sequential correlations. Recurrent neural networks fail to derive features in parallel, but convolutional networks can learn local responses from dimensional data. Consequently, the amalgamation of the two networks gives us both the benefits from the two and allows us to predict circRNA more accurately. Our method completely eclipsed the baseline models when predicting circRNA. It achieved 0.8933 accuracy and 0.7730 MCC on test data. We compared also the performance of all methods described above using 5-fold cross-validation on training data, and the results are shown in supplementary table S6. We achieved almost identical results using cross-validation and also when we ran validation and test data, which suggests that we have a low risk of overfitting by using validation data and an early stop strategy. 3.3.2 Model analysis. In order to analyze the aspect of our architecture used to extract the ACNN BLSTM descriptor, we evaluated its robustness and interpreted the influence of 4 hyperparameters: the k-mer length k, the splitting stride s, the Maxlin parameter and the embedding. Figure 5. A. Pearson's correlation coefficient as a function of HK, L0, score for different k-mer length k and different flaking sequence length L0. B. Pearson's correlation coefficient as a function of VK, L0, Score for different k-mer length k and different flaking sequence length L0. Table 1. Performance comparison between our ACNN BLSTM proposed architecture and other baseline methods for sequence. Classification method train val test #epochs time. ACC MCC 1 hot CNN 0.8176 0.8185 0.8117 0.6164 24 368 ACNN 0.8402 0.8385 0.8330 0.6625 32 308 CNN 0.8225 0.8208 0.8194 0.6299 38 338 LSTM 0.7836 0.7820 0.7826 0.5568 24 712 BLSTM 0.8029 0.8034 0.8020 0.5997 31 748 Glove CNN BLSTM 0.8804 0.8756 0.8737 0.7324 48 354 1 hot CNN BLSTM 0.8609 0.8570 0.8541 0.6890 42 394 ACNN BLSTM 0.8947 0.8935 0.8933 0.7730 36 347 78 M. Chaban et al. Dimension D. We evaluated our model using the MCC measure on the test set, and the results are shown in Figure 6. Starting with the k-mer length, k, we expected that increasing k would increase the total number of different k-mers, increase the information in the co-occurrence statistics and improve our model. However, we find that k1/3 gives the best performance. In fact, as the k-mer size increases, the performance decreases. This can best be seen in the significant drop in performance at k1/5. This leads us to the importance of mining the sequences with trinucleotides for circRNA's prediction. Even though circRNAs are classified as non-coding RNAs, recently, several studies demonstrate that circRNAs can be translated. Legnini et al., 2017, Pamu-Doherty et al., 2017, Tatimer and Willis, 2017. In agreement with these findings, the peak at the k-mer size of 3, which is the size of one amino acid, in Figure 6a suggests that the translation of circRNAs is not trivial. The second hyperparameter is splitting stride, S. As expected, a larger stride will decrease the corpus size, which in turn, decreases the contained information in the sequence, and decreases the performance. Therefore, we set stride at S1/1 in our model. The third hyperparameter is maximum sequence length, max-len. There is a positive correlation between MCC and max-len. The reason behind the correlation is obvious, as some input sequences are truncated at shorter lengths, which results in a loss of information contained in the sequence. We were limited to a max-len of 8,000 basis points, due to limitations in computer memory. The final hyperparameter is the embedding dimension D. We can see that it has no impact on the performance of the model. However, when D is increased, the time to train the weights of the embedding matrix is increased. This led us to choose D1/40 in our model. Next, we investigated the impact of different filter configurations in the asymmetric convolutional layer on the model performance. In Figure 7, we show that the MCC measure on the testing data using different filter configurations. For each filter configuration, the best result under extensive grid search on hyperparameters is shown in Figure 7. In the asymmetric convolutional layer of our model, filters are utilized to represent local trinucleotide features. We initially thought that several convolutional layers in parallel with filters of various sizes would perform better than any single asymmetric convolutional layer with filters of the same length due to different sized filters being able to find features of different n-grams, n consecutive 3 mers. However, our experiments demonstrated that a single convolutional layer with a filter length of 7 always surpasses the other options. When multiple layers were run together, filter combinations with a filter of length 7 always performed better. This further aided the verification of 7-gram features playing a vital role in representing local features while predicting circRNA. 7 grams means we extract local features from every 7 overlapping trinucleotides, which means 3 non-overlapping trinucleotidae. This is in agreement with the results found in a study, Asghari and Mofrad, 2015, where using the word 2-vec skipgram model was able to generate trigram of amino acids representations. One amino acid is equivalent to trinucleotide in DNA sequence, reproducing known physical relationships that were useful for protein classification. Our results show once again how circRNA sequences share characteristics with coding sequences when mining the sequences with text mining techniques where the sequence is treated as collection of k-mers. 3.4 Feature fusion performance for classification of circular RNA from otherlong RNA. Table 2 shows the performance of unimodal classifiers, bimodal classifiers, multimodal classifiers and circRNA. For unimodal classifiers, we stack 2 fully connected layers for each descriptor. For bimodal classifiers, we fuse 2 descriptors with each using our feature fusion approach. For multimodal classifiers, we fuse all 3 descriptors using feature fusion fine-tuned method described above, feature fusion by just the concatenation of the representations from individual trained DNN per descriptor without the tuning operation, early fusion and late fusion. See supplementary table S7-4. Figure 6. Matthews correlation coefficient, MCC, scores for test data with varying 4 hyperparameters. Figure 7. Matthews correlation coefficient scores for test data with different filter size strategies. For the horizontal axis, S means single convolutional layer with the same filter length and M means multiple convolutional layers in parallel with different filter lengths. In both single and multiple filter lengths, a filter of length 7 always showed higher MMC score, blue bars, table 2. Performance comparison between unimodal classifiers, bimodal classifiers, multimodal approaches and circRNA accuracy. MCC F1 score time, min, sec 0.8977 0.7792 0.8596 0.2 cons 0.7611 0.4969 0.689715 RCM 0.7158 0.4072 0.6427 sec Thorn RCM 0.9263 0.8520 0.926847.2 sec Thorn cons 0.9333 0.8654 0.9333 15.2 RCM Thorn cons 0.8478 0.6838 0.810262 feature fusion fine tune 0.9417 0.8833 0.940262.2 feature fusion 0.9325 0.8653 0.932162.2 early fusion 0.9271 0.8542 0.927862.2 late fusion 0.9314 0.8628 0.930762.2 circRNA 0.8056 0.6113 0.8108 515 sec. ACNN BLSTM sequence descriptor, cons, conservation descriptor. RCM descriptor, time needed to extract features used for classification for test data. Bold ones are the best performer in each column. circRNA classification via deep learning 79. Performance comparison using 5-fold cross-validation on training data. We also compared the time needed to extract all features on the test data, which contains 7,630 samples. The comparisons were performed on an IBM desktop with 8 Intel Xeon CPU cores at 2.33 GHz and 24 GB memory. As indicated in Table 2, ACNN BLSTM sequence descriptor outperforms 2 other descriptors with an improvement of more than 15% in F1 score, which reflects its huge importance for our task. Even though our proposed RCM descriptor had the lowest performance capability with a high running time to be extracted, it was considered good with a 0.64 F1 score. We can also see its effect in improving performance when combined with other descriptors. It is interesting that only 0.2 minutes is required to extract the ACNN BLSTM descriptor within 0.8596 F1 score, while circRNA required approximately 8 hours and 35 minutes to extract all features. This included graph features, component composition features, including frequencies of trinucleotides, conservation score features, features of ALU and tandem repeats, the ORF and SNPs from transcripts. Graph prod is taking most of running time, with only in 0.8108 F1. This reflects the importance of sequence patterns and long-range dependencies between trinucleotides in predicting circRNAs. As expected, Table 2 shows that the multimodal fusion produces much higher performance than any individual modality. This indicates the strong complementarity shared between the 3 descriptors. Furthermore, feature fusion fine-tuned, which is adopted by circDeep leads to a 0.9402 F1 score. This is greater than all unimodal learners and also other multimodal fusion baseline methods. To once again make a comparison with circRNA and HELM, we tested our tool with the data proposed by Pan and Xiong 2015, and the results are shown in Figure 1. This further supported our results and interpretations by having an improvement of more than 12% in accuracy using our proposed method. For conclusions in this paper, we propose a new multimodal deep learning tool circDeep to distinguish circRNA from otherlong. Compared to existing approaches, our approach has the following advantages. It takes advantage of our proposed RCM descriptor, which can provide the likelihood of circularization given the flanking sequences and the query sequences. 2. It successfully integrates the ACNN, BLSTM and DNNs for dealing with the different input data types. 2. Enhanced discrimination ability. 3. The hybrid framework of flexible multimodal learning and fusion at an abstraction level makes our approach handle different features in an easy manner. The top shared hidden layer at the fusion level will improve discovery of shared properties across different modalities. 4. circDeep achieved an improvement of more than 12% in accuracy in both datasets with a very small running time compared to existing tools. Acknowledgements. We would like to thank Eric Rushka and Julia Koreiker for insightful discuss science and comments. Funding this work was supported by the CPRN Investigator Development Award, IDEA, from the NIH National Institute of General Medical Sciences, P20GM103436, and NIH NIGMS Area Grant, R15GM126446. Conflict of interest, none declared. References. Asghari, E. and Mofrad, M.R., 2015, Continuous Distributed Representation of Biological Sequences for Deep Proteomics and Genomics. PLOS 1, 10, E0141287. Bachmeier Hayda, A., et al. 2015, Correlation of Circular RNA Abundance. With proliferation exemplified with colorectal and ovarian cancer, idiopathic lung fibrosis, and normal human tissues. SciRep. 5, 8057. Bird, C.E. et al. 2010, Expression of linear and novel circular forms of an INK4/ARF associated non-coding RNA correlates with atherosclerosis risk. PLOS Genet, 6, E1001233. Chen, G., et al. 2012, Lung-Carnet disease, a database for long non-coding RNA-associated diseases. Nucleic acids Res, 41, database issue, D983-D986. Chen, L., et al. 2015, Circular RNAs in Eukaryotic Cells. Kerr. Genomics, 16, 312-318. Chen, L., et al. 2018, Discriminating CRNAs from otherlong using a hierarchical extreme learning machine, HELM, algorithm with feature selection. Mol. Genet. Genomics, 293, 137. Cooper, T.A. et al. 2009, RNA and Disease. Cell, 136, 777-793. Darian, T., et al. 2012, The GenCode V7 Catalog of Human Long Non-Coding RNAs, Analysis of Their Gene Structure, Evolution, and Expression. Genome Res, 22, 1775-1789. Dubin, R.A. et al. 1995, Inverted repeats are necessary for circularization of the mouse testis retranscript. Gene, 167, 245-248. Erickson, M., et al. 2003, Recurrent de novo point mutations in lamin A cause Hutchinson-Gilford progeria syndrome. Nature, 423, 293-298. Goldberg, Y. and Levy, O. 2014, Word 2 Vec explained, deriving Mikolov et al.'s negative sampling word embedding method. R14 preprint R14, 1402.3722. Hall, M.A., 2000, Correlation-based feature selection of discrete and numeric class machine learning. In Proceedings of the 17th International Conference on Machine Learning, pages 359-366. Hansen, T.B. et al. 2013, Natural RNA circles function as efficient microRNA sponges. Nature, 495, 384-388. Ivanov, A., et al. 2015, Analysis of intron sequences reveals hallmarks of circular RNA biogenesis in animals. Cell Rep. 10, 170-177. Lasda, E. and Parker, R. 2014, Circular RNAs, Diversity of Form and Function. RNA, 20, 1829-1842. Legnini, I. et al. 2017, circZNF609 is a circular RNA that can be translated and functions in myogenesis. Mol. Cell, 66, 22-37E29. Lukiu, W.J., 2013, Circular RNA, circRNA, in Alzheimer's disease AD. Front. Genet, 4, 307. Matic, J.S. and Makunin, IV, 2006, Non-coding RNA. Hum. Mol. Genet, 15, R17 R29. Mumzak, S. et al. 2013, circular RNAs are a large class of animal RNAs with regulatory potency. Nature, 495, 333-338. Mercer, T.R. et al. 2009, Long Non-coding RNAs, Insights into Functions. Nat. Reverend Genet, 10, 155. Mikolov, T. et al. 2013, Efficient Estimation of Word Representations in Vector Space. R14 preprint R14, 1301.3781. Min, X, et al. 2017, Chromatin Accessibility Prediction via Convolutional Long Short-Term Memory Networks with K-MER Embedding. Bioinformatics, 33, I92 I101. Morel, CF et al. 2006, a LMNA splicing mutation in two sisters with severe Dunnigan-type familial partial lipodystrophy type 2. J. Klin. Endocrinol. Metab, 91, 2689-2695. Amudurti, N.R. et al. 2017, Translation of circRNAs. Mol. Cell, 66, 9-21E27. Pan, X. and Xiong, K. 2015, PreCircRNA, Computational Classification of Circular RNA from Other Long Non-coding RNA using Hybrid Features. Mol. Biocyst, 11, 2219-2226. Shrivastava, N. et al. 2014, Dropout, a simple way to prevent neural networks from overfitting. J. Mach. Learn. Rees, 15, 1929-1958. Sejadi, C. et al. 2015, Going Deeper with Convolutions. In 2015 IEEE Conference on Computer Vision and Pattern Recognition, CVPR. Pages 1-9. Katimer, D.C. and Willis, J.E., 2017, An Uncharted Journey for Ribosomes, Circumnavigating Circular RNAs to Produce Proteins. Mol. Cell, 66, 1-2. Zheng, H. et al. 2016, Convolutional Neural Network Architectures for Predicting DNA Protein Binding. Bioinformatics, 32, I121 I127. Zhang, X. O. et al. 2014, Complementary Sequence-Mediated Exon Circularization. Cell, 159, 134-147.